Spermatogenesis and oogenesis both involve mitosis, cell growth, two divisions of meiosis, and differentiation. Mitosis is the creation of germ cells, not the ones that you kill with hand sanitizer, but gametes, aka sperm and eggs. First, we're going to talk about oogenesis, which is the creation of egg cells within the ovaries of the uterus. So, technically, oogenesis is only meiotic events, which can be seen on the left-hand side, but we decided to pair it up with follicle development to show you how a period works. This whole process begins before birth. On the left, we have oogonium, which is the stem cell, and on the right, we have the primary oocyte, aka the primordial follicle. Via mitosis, the oogonium becomes an egg. Then, when the oocyte becomes 4 to 5 months old, it and its surrounding cells begin to divide via meiosis, which will eventually stop at prophase 1 when birth occurs. Once again, the egg cell will grow. This red line represents childhood. During this time, the ovary is inactive and nothing occurs. From puberty to menopause, every month the following events take place. Follicles are created and stored in the ovaries from birth, but with each menstrual cycle, a new primary follicle will be stimulated by follicle-stimulating hormone, better known as FSH. On the left side, you'll see our gamete attached to a diamond. This diamond is the mitotic spindle, which separates the chromosomes into daughter cells during mitosis. The first division of oogenesis creates one big cell, and one small cell. The small cell is the first polar body, which will eventually die off. The larger cell undergoes a second division of meiosis, which is later completed post-fertilization. And here you will see this is the second division. Again, one large cell and a small cell are created. The small cell is known as the second polar body, which will also degenerate and die. The larger cell is now a female gamete, which survives. The egg gamete is much bigger than the sperm gamete. That's why we put them both together here. And the rest of the follicle development results in a period and the releasing of a gamete. Secondarily, we are going to talk about spermatogenesis, which is the creation of sperm within the testicles. Within the testicles, there are seminiferous tubules, and the gaps between them known as interstices. These tubules almost look like a plate of spaghetti. So the outer layer of the cell is called a germinal epithelium, the wall of the seminiferous tubules. And that's where all the production begins. As cells mature, they migrate into the tubules. The cell wall has Sertoli cells, which is the diamond right here, that facilitate the production of spermatozoa. Here's a close-up of the seminiferous tubule. So first off, we have the germinal cell, which is diploid. It is also present in the close-up photo, which is the closest one to the yellow cell wall. After a little growth, we have the primary spermatocyte, which can be seen on both diagrams. On the left, it can be found below the germinal cell. Next, via meiosis 1, our primary spermatocyte becomes 2 secondary spermatocytes. As you can guess, it can be seen on the left photo, right below the primary spermatocyte. Meiosis 2 then occurs and creates 4 spermatids. On the left, they are represented by little sperm, which will later on grow to become spermatozoa. Spermatogenesis, unlike oogenesis, occurs 24/7 after puberty, and all stages of sperm can be seen in the body. Next, we're going to talk about fertilization. But before we do, we have to kind of explain a little bit about spermatozoa. It contains one haploid nucleus, a way to move, and an enzyme complex so it can penetrate the egg. All of this can be seen on the diagram. The acrosome, which is the little yellow bit, contains the enzyme complex to penetrate the egg. The nucleus, which is also known as the brain, and the flagellum is the tail so it can move. Sperm have receptors to detect chemicals excreted by the egg in order to find it. When the egg is initially reached, there are specific steps that will result in the union between sperm and egg. Steps are taken to prevent polyspermy. Polyspermy is when an egg is fertilized by more than one sperm, which can lead to many defects. The first step is the acrosome reaction, where the zona pellucida, a layer surrounding the egg, and the acrosome bind together. The acrosome enzymes are released, digesting the zona pellucida. The second step is penetration of the egg membrane. The previous step exposes the acrosome used to bind to the egg membrane, and then the first sperm to penetrate through the zona pellucida and connect itself will have its nucleus enter the egg and complete fertilization. The last and most important step in the cortical reaction is once sperm and egg are binded, the egg is activated. This will set off cortical granules located near the surface of the egg membrane to release their contents via exocytosis, which will then digest the remaining sperm. This will prevent polyspermy and harden the zona pellucida. Alright, so the prevention of polyspermy can be explained by this wonderful reenactment. So the Earth will represent an egg. Aliens, aka sperm, have receptors to sense our presence and will find the egg. Once one alien penetrates our atmosphere and comes to Earth, the Incredibles, our cortical reaction, will set up a force field to kill the rest of them because we don't need all the aliens. We only need one to show the world that aliens are real. Next, we're going to talk about internal versus external fertilization. Fish and aquatic animals, just like salmon, commonly use external fertilization by releasing their eggs into the water so fertilization occurs outside the female. There are plenty of risks with this method due to predation and environmental variations, but it allows more genetic variation and offspring. Internal fertilization, on the other hand, is generally used by terrestrial animals that live on land. Otherwise, their gametes would become like dry little raisins. This method of fertilization ensures that sperm and egg are close to one another, so fertilization is more lucky. Some marine animals also use this method of fertilization. The embryo can be protected inside the mother and help the species become more selective in their partners. Unfortunately, this requires intimate contact, which can lead to STDs. And due to the small internal room in most of these animals, it can also limit the number of offspring reproduced.